This next assay that we are going to talk about is enzyme-linked immunosorbent assay, or ELISA for short. This is a quantitative immunoassay. So enzyme-linked immunosorbent assay, it's used to detect presence of antigens again in biological samples using antibodies to target the exact one that we are looking for. We want to use very highly specific antibodies for this one to minimize cross-reactivity and to get better quantitative data. We can use specific types of monoclonal antibodies that are targeting a particular part of the antigen. So maybe, just the amino terminal or the carbohydrate recognition domain, whatever it is that we really care about within that protein to try to make it as specific as possible. It could even be against a phosphorylation state or an active state of the protein. So some terminology for ELISA. You need to have some type of solid phase that we are going to use for the assay. This is usually a microtiter plate. This could be in the shape of 8 by 12 rows. 8 rows of 12 wells, or a typical 96 well plate even. If you have a 96 well plate, obviously you can lose a lot of wells if you're not using them all at the same time, if you don't treat them properly. So sometimes having just rows of 12 wells each helps, because then you can just utilize as many as you need and not waste any. Then you have this term called adsorption. This is the process through which we either bind the antigen or the antibody to that solid phase that we are using, so at the bottom of the well, by incubating it with it under specific conditions in a buffer so that it can attach to that solid phase. Then you have your antigen, obviously, which is going to elicit the production of antibodies. So that's the protein of interest in our case that we are going to care about, or the part of protein that we care about. And then our antibodies are going to be the ones that are going to be produced, and that is what we're going to be utilizing to bind to our substance, our samples, to look for our protein of interest. And then we have this term enzyme conjugate. This is when you attach the enzyme irreversibly to an antibody. And this is usually going to be our detection molecule, so that we can detect for it and then solubilize it and quantify it. This could be horseradish peroxidase, HRP, alkaline phosphatase, AP. These are the two main ones that we use. And then finally, we have that color-changing compound, chromogen. This is the chemical that's going to change the color when it binds to that enzyme and interacts with it. And that substrate change, the color reaction that we get, is what we're going to be quantifying through our spectrophotometer. So an example of that would be trimethylbenazidine, TMB, which is a common one used in ELISAs. So here you have an example where you have your well of the solid phase that we're going to be using. Your antigen or your antibody is going to be what's adsorbed to this plastic surface. That's what gets you that sorbent, immunosorbent. Antigen, because we are recognizing the specific antigen using an antibody, that's why it's called immuno. And then the antibody that is recognized and used for enhanced is going to be our second antibody. That's another antibody. Just like in Western, we used multiple antibodies. One primary antibody against the antigen, and then a secondary antibody to enhance the signal or to produce that color reaction. This secondary antibody is what's usually enzyme linked. And then you have your substrate that's going to react with that, whatever that enzyme is that is attached to your secondary antibody, to produce the color product that we are going to detect and quantify. So there are many different types of ELISA, and they could be on the basis of detection and also on the basis of how we perform the protocol. So on the basis of detection, we can have colorimetric, chemiluminescent, or competitive fluorescent. Obviously, in a chemiluminescent ELISA, you're going to have a light reaction, similar to the luminol we have in Western blots as well. You can have chemiluminescent there as well. In a colorimetric, you have the color change with our chromogen, the substrate that binds to the enzyme, interacts with it and causes the change of color that is then examined. And then in competitive fluorescence, you would have some kind of a fluorescence change as the result. On the basis of the protocol or the method that is utilized, there are non-competitive and competitive ELISAs. Within the non-competitive, there are three main ways that we can examine our for our protein of interest. You have direct interactions, a direct ELISA, indirect ELISA, and sandwich ELISA. In a direct ELISA, we coat our solid phase with our actual samples, our proteins extracts that we have. We then bind a primary antibody that is specific to the antigen of interest that is already conjugated to an enzyme that is going to cause the color change. And then there is no other step. We basically just bind the antibody, wash it off, wash off anything that's not bound, and then add the chromogen to cause the color change with the substrate, and then that color change is what we're utilizing. This is great if your protein of interest is present in a large amount and there is very clean sample, there are not a lot of proteins, and also if that protein of interest has good binding capacity to your solid phase. Otherwise, it could be a rough going. To try to enhance that signal further, we can do an indirect ELISA where we utilize again our antigen that is bound to a primary antibody that is specific to the portion that we are interested in, then a second primary antibody conjugate that is going to contain the enzyme, and then you can again add your substrate and get your color change, and that's what you are going to quantify. This is going to enhance your signal a little bit and help you see it a little bit better. Many times, direct as well as indirect ELISAs are limited to proteins where you have a lot of them, frankly, or to simple mixtures or simple samples. When you have complex systems like your cell extracts that have thousands of proteins, lots and lots of them with lots of background, essentially, it is going to be really hard to get your protein of interest to work very well with them. In those cases, what instead we want to use would be something called sandwich ELISA. In a sandwich ELISA, we coat the bottom of our wells, our solid phase, with an antibody, what we call a capture antibody. This antibody is going to be against the same protein of interest that you're interested in. So galectin-3, for example, if I'm interested in galectin-3 and quantifying its amount, I could use a capture antibody against galectin-3. This could be a monoclonal antibody, it's just a general antibody, doesn't matter. Usually it is a monoclonal, just to simplify the system, and it could be to any part except the one that I care about, because we are going to use a second primary antibody after adding my protein extracts. So you add the captured antibody, you coat the surface with it, you cross-link it to your plastic surface. That can be done in advance. You can seal them, you can put them in the fridge, and you can use them up to a couple of months later. You would then add your protein extracts that you want to look at, that you want to examine on top of that, and you're going to allow it to bind to your capture antibody. You're going to let that happen for a couple of hours minimum. You could do overnight, but typically we just want to do 1 hour, 2 hours. That's good enough. Then you wash away anything that did not bind. So this is going to simplify your system, because only my galectin-3, your protein of interest, should bind to that capture antibody. Now you're going to come in with a second primary antibody against galectin-3 that binds to a different epitope than the capture antibody. So it can bind to another place on there. This is going to really this is why it's sandwich, because you have an antibody and an antibody on the bottom and an antibody on top. This is going to really make that binding strong in this case. And now the second antibody is going to be binding to hopefully the epitope. If you have a nice clean epitope that you're looking at, it's going to bind to that portion of your protein. You then use a second antibody. This is your secondary antibody that contains your enzyme-linked conjugate. And then you add the substrate and you get the reaction, the interaction, the color change that you can quantify. This is going to be super specific and allow you to get very nice, clean, quantitative results. These are the type that we are going to try to do for CREB in our class. Another way that we can look at it is what we call a competitive ELISA, where you use an inhibitor antigen that is going to work with yours to show how much of your antibody is bound to your specific protein compared to the inhibitor antigen. And that's another way that we can examine it as well. So in a direct ELISA, just to remind you, antigen is directly immobilized to the assay plate. It requires that you have lots and lots of this. It is very cheap, relatively cheap as ELISAs go, because you just have a single antibody. It's primary labeled antibody. It's going to act with your antigen. It's less sensitive, obviously, because it's not going to be very specific. And it is not widely used for ELISA purposes, but in immunohistochemical staining, where you have cells and tissues, it can be used very well. Indirect ELISA is going to be a little bit more sensitive. It is going to utilize an unlabeled primary antibody and a secondary antibody. That again, it could be that general secondary that we had before, as long as it has an conjugate on there that we can use to enhance the signal. The secondary is going to enhance the signal. The conjugate is going to give you the color change when the substrate does it. And then the sandwich ELISA, which is the one that we are going to be doing, it is very helpful when your protein of interest is in low abundance within a very complex mixture, or just, you don't have a lot of it to work with and you are looking for a very specific portion. So antigens like tumor markers, hormones, serum proteins, these are going to be best quantified using sandwich ELISA. So you're going to have your capture antibody against your antigen of interest bound to the solid phase. Then a second primary antibody, again, this does not need to be labeled, it's going to be unlabeled, could be the same as your Western, or specific to a different epitope that you're interested in. You wash away, in each step, you wash away any unbound sample, and then wash away any unbound antibody. And then you use a secondary antibody with your enzyme conjugate that can react with the substrate and give you the color change. So some troubleshooting problems that can happen. If your negative control, so if you had no protein present and you're getting a positive result, the most likely cause of that is going to be contamination of your substrate solution, or maybe you didn't block your plates. Similar to Western, you have to block plates in between to remove any background noise. If that isn't done, or if you were in a hurry and didn't wash your wells very well with appropriate buffers, that could also cause negative controls to give some positive results. If you don't have any color in any of your samples, including your positive controls, you obviously want to check all the dates for all this and storage conditions for all your chemicals. But it could also mean that your microwell plates were not coated properly, either with the antigen or with the antibody, or you maybe applied them in the wrong order. So if you had the capture antibody and you put the primary before you added your sample, it's not going to work very well. So you always want to make sure that you can follow the steps very well in an ELISA, because it does have a bunch of steps and it does take time. If you have very little color that develops in your positive controls or your test sample, again, you want to make sure that you are diluting your samples appropriately, and then as well as your reagents appropriately. But also many times, and especially with student samples, most of the times it's because of not removing the wash buffer completely, because if you don't drain all the wash buffer, you are diluting essentially your samples, your antibodies, more than what you think, and that is going to give you less than appropriate results. And then also, if you have test samples that are developing color but your positive controls don't, because our positive control is going to be pure protein that is source, you want to make sure that it is not it is stored properly and that it is not expired, because they do degrade over time. It's a pure protein. It is going to lose its just structure over time. And finally, if you can see it but you can't actually see it with the numbers and absorbances. The biggest reason for that is to make sure that you are using the right wavelength on the spectrophotometer. So I hope this helps to give you a little bit of an overview on ELISAs and how we are going to be using them, and gives you an idea on how to troubleshoot any common issue.